Okay, right, I think we've got everyone, and everyone has now successfully joined the Hangout. Thanks for coming, everyone. So we're here today to chat to last year's participants of eLife's Early Career sponsored presentation series, and also to announce this year's participants at the end of the Hangout. eLife have started this series with the aim of helping out the early career researchers that publish with us, because we know there's a lot of pressure involved at this stage in a researcher's career. To this end, we've recently sponsored 4 researchers who are early on in their careers to present their work at any one of our founding organisations, the Howard Hughes Medical Institute, the Wellcome Trust and the Max Planck Society. 3 of the recipients are with us today, and they're ready to talk about their work and experiences as early career researchers. The discussion today is going to be moderated by Dr. Chris Smith. Chris is a medical consultant specialising in clinical microbiology and virology at Cambridge University and its teaching hospital. He's also the founder of the Naked Scientists, a media-savvy group of physicians and researchers from Cambridge University, who use radio, live lectures and the internet to strip science down to its bare essentials and promote it to the general public. So without further ado, I think we'll pass over to Chris, who will talk a little bit more about what he does at the Naked Scientists, and he'll introduce the 3 speakers for us. Chris, away we go.
Thank you, Dennis. Good afternoon and welcome to this eLife Hangout, in fact one of my first official Hangouts. I'm very pleased to be joined by 3 early career scientists from eLife who have published recently in the journal, or at least over the last 12 months. My name's Chris Smith and I am the managing editor and founder of The Naked Scientists, and I also co-present that program, which is broadcast internationally on both terrestrial radio and also over the internet. And when I'm not doing that, I work at Cambridge University's Teaching Hospital Addenbrooke's, where I'm a consultant virologist. So I'm interested in the viruses that cause infections in humans or pose a threat to humans. Now, the idea of this Hangout is to have a conversation with 3 individuals who are fairly well, you could be described as high achievers in their careers so far, find out a bit about their careers and where they're hoping they're going to go, and then take questions from anyone who may come in with those questions. We hope that some people will join us as we go through. But let me outline for you who those individuals are. So Jesse Bloom is an assistant member at the Fred Hutchinson Cancer Research Center, that's in Seattle, and prior to beginning his faculty position, Jesse obtained his PhD in chemistry and also did a postdoc in biology at Caltech. His lab uses a combination of experimental and computational approaches to study influenza evolution, and his paper was published with eLife looking at mutations in the nuclear proteins of human H3N2. We're also joined by Wenhui Li, who received his PhD from the Peking Union Medical College and Chinese Academy of Medical Sciences back in 2001. He worked as a postdoctoral fellow at the Department of Microbiology and Molecular Genetics at Harvard, and that was with Dr. Michael Farzan. And in 2007, he joined the National Institute of Biological Sciences, NIBS, which is in Beijing, as an assistant investigator. He's interested in molecular mechanisms of viral infection. I was very lucky to interview Wenhui when he published his paper discovering the entry receptor for hepatitis B. And hepatitis B is a major global threat with at least, I think probably in the region of 2 billion people out of the world's 7 billion people, either latently, chronically, or previously infected with hepatitis B. It's probably the most common virus that can establish a chronic infection that we know of and a major health threat. Also, Rosie Alegado is with us. She's an assistant researcher at the University of Hawaii, Manoa, and is faculty in the Department of Oceanography and the Sea Grant College program. Long-term goals in her research are to understand how chemical cues from bacteria shape the host microbe evolutionary pathway. Her work's motivated by the recognition that eukaryotic life came out of the seas teeming with bacteria, and that those microbes continue to play very critical roles in evolutionary, biological and geochemical processes. The paper that Rosie and I discussed, which she published about a year ago, explored how the morphology of an organism called Salpingoeca rosetta, and you can correct me, Rosie, how I say that properly, it's actually a choanoflagellate, how that gets influenced in terms of its structure by its interactions with various species of bacteria. So let's start with Jessie, though. So Jessie, tell us a bit about you first. I've given the potted history, but what actually are you looking at in relation to how flu does its thing?
Yeah, so in my lab, we're especially interested in, so for example, the paper that we published in eLife was sort of attempting to look at how interactions between mutations help shape the evolution of influenza. So in general, when you think about evolution for any given viral sequence, there's a somewhat limited number of single mutations you can make to the virus, but the complexity comes when one of those mutations can then influence impact of a future mutation. So that can make evolution very complex and unpredictable. For example, you can have a mutation which doesn't have any immediate effect, but then enables the virus to acquire subsequent mutations that have a lot of phenotypic relevance. So we're interested in that phenomenon and also sort of understanding additional evolutionary patterns in influenza evolution. And so a lot of the approaches we take in my lab are to look at natural influenza sequences and try to identify patterns and commonalities in the evolution, and then use that to generate hypotheses that we test in the lab.
So if one looks at how flu makes its global journey that it does every year, would the aim of your work be to therefore say, well, if we look at flu in Australia, where it's currently coming up towards winter time now, which is probably going to be the flu that comes up to us in the northern hemisphere next year in 6 to 8 months' time, if we look at some mutations which are happening there now, those may beget other mutations that will surface by the time the virus completes its journey and comes back to us.
Yeah, I mean that's definitely true. I mean I don't think that predicting the specific mutations that are going to arise or spread is an entirely tractable problem. We can make some progress predicting maybe the evolution over very short terms by looking to see what's out there. But evolution is both a stochastic process and a very complex process. So I guess a lot of the focus of what we've been working on is less trying to predict what the specific mutations are going to be and more trying to understand the constraints and the patterns. And a lot of this is motivated by sort of interest in questions in evolutionary biology as much as specific questions about influenza per se. I mean, the really great thing about influenza, and the reason I actually originally became interested in it, is because most of the time when you look at things like in evolutionary biology, you have very large amounts of divergence between what you're looking at, and sort of the current extant species, but you don't know what happened in the evolutionary past. And so for influenza, for example, if we look at the viruses that are present today in humans and pigs that have diverged in just the roughly 90 years since 1918, those viruses are actually as different at the protein level as a human and a mouse. So it's pretty amazing in the sense that we can see this much evolutionary divergence, and we can really look mutation by mutation to understand what happens, versus the standard situation in evolutionary biology when you have two things that are very different and you don't have a lot of insight in terms of how they got to where they are. So although I think some of our research does have some applications to public health and sort of these medical virology areas, a lot of what we're focusing on, and for example what we're working on in a paper in eLife, is to really understand these evolutionary dynamics and understand how mutations interact to enable the virus to sort of acquire phenotypically relevant mutations, such as mutations that promote immune escape.
Does it become more complicated because flu has what we dub a segmented genome? It's a bit unusual in the virus world because the genetic material comes in little packets or nucleoproteins, and they're all independently replicating in a cell, which means you can.
I would say in certain ways that, from the analysis perspective, in some ways it almost makes it easier. So almost all viral genomes, almost all most genomes in nature recombine in some form or another. So if you're looking at, for example, a human genome or an HIV genome, you have these genomes that can sort of recombine at, in principle, any point along the genome. And so for flu, as you were describing, the genome is in segments. And within those segments, there are 8 gene segments. Within those segments, the rate of recombination is 0 or very close to 0. But then the segments themselves recombine very efficiently, and the process is actually called reassortment because it doesn't really require any breaking of the nucleic acid. So it's sort of a very efficient form of exchange that sort of equivalent to homologous recombination. And I would say actually, I mean, that certainly means that there's a lot of that sort of recombination, but in a certain sense from evolutionary analyses it makes it simpler because you know what parts of the segments are going to be sort of inherited without recombination, and then you know that the exchange happens, you know, between these segments. So I would say from a practical standpoint it makes it easier for us, but it certainly means that influenza has a very high rate of reassortment, and these segments are fairly modular such that fairly diverse strains of influenza can exchange segments. So certainly this is a big contributing factor to the emergence of new influenza pandemics, the fact that these viruses can exchange genetic material very easily.
Well, before we move on to another virus, which is hepatitis B, before we all get maxed out on viruses, let's go, I suppose you could say, a rose between two thorns. Let's talk to Rosie. So, Rosie, you and I spoke last year when you were working on these fabulous organisms, which as soon as they get a bacterium to snuggle up to, they have this dramatic change in shape. Can you tell us a little bit about them, and not least how we pronounce the name of them properly?
Right, so choanoflagellates, the word is kind of a compound. So choano means collar, and flagellates obviously refers to the flagella. And so these are unicellular organisms that can transition between unicellular and multicellular states. And they swim around in the water column, so they're aquatic, found both in marine and freshwater environs. And they're very useful because they're the closest unicellular relative to animals. And so the fact that they can transition between a completely one-celled state and, you know, temporarily a multi-celled kind of structure really lends itself to asking the question about how some of the molecules, such as the proteins that they have, how that might have evolved into the toolkit for animal multicellularity.
And how can you unpick that process by looking at them?
Well, so obviously you can do a lot of cell biology work and kind of look at the exact mechanism and steps and processes of how that occurs. And so work by other people in Nicole King's lab, which is where I did my postdoc at UC Berkeley, has really kind of begun to tease apart what are the steps involved, at least in Salpingo rosetta, or S. rosetta as we call it in lab, that process. And so we know that it's a process of incomplete cytokinesis, so that you have a single founder cell that then divides and remains together, and that those cells are held together by fine intracellular bridges, which is very much homologous or analogous to what we see at the very early stages of animal development. Nicole King has also, while I was in her lab, we sequenced the genome of S. rosetta. That was a graduate student, Stephen Fairclough. That work came out in genome biology. And most recently, work by a really amazing graduate student, Tara Levin. She was able to begin to do genetics in the choanoflagellate S. rosetta. And I think that that really, in combination with the ability to look at the transcriptome, is going to allow us to open up that field hugely and to look at what the genes, at least on the choanoflagellate side, that are involved.
So given that we're a bit more complicated than they were then, the earliest life, I mean,
yeah.
How do you think this process actually evolved in the first place to become what we have today as us?
So it's my hypothesis and our hypothesis in the King Lab that we like to throw around, that the very processes that were involved in these transient transitions while we were while we were unicellular were perhaps co-opted or repurposed for life as a multicellular organism.
And then cells gathered that it would be quite useful to stick around together. Yeah. And so you ended up with metazoal life rather than unicellular life.
That's right. So the transient transitions eventually became co-opted into a more permanent lifestyle. And so rather than being facultative, it was kind of a requirement for all the time life, for life all the time, rather.
And can you see that genetically in the genome history that you're performing? So
for the choanoflagellates, you can certainly see that this group of organisms, and now it's beginning to be the case for a little bit more distantly related organisms as well. Other groups are kind of beginning to sequence these really odd protists or protozoans that are kind of like our nearest cousins. What we're seeing is that sets of genes that were normally thought of as being very animal specific are actually present, or at least the remnants of these, or let's say the building blocks of these, the protein domains, were present actually before animals became animals. And so perhaps when along that stem path to the evolution of what eventually became our ancestral animal, what we think might have been happening, at least one mechanism, is that you have protein domains that were separate in unicellular organisms that then coalesced and then, through genome shuffling or protein shuffling, kind of came together, and then you had the combination of new functions or the adoption of kind of new functions towards life as an obligately multicellular organism.
So what do you think actually came first in that process?
What was the first thing that happened? That is extremely hard to say. But I think the first thing that probably came about was either the ability to stick together after dividing, or perhaps the ability to sense the environment. So I mean, when you think about the classic pathways, the developmental pathways, people think of generally about 7 really, really classic animal developmental pathways. And we see that there are a number of those pathways are present in quantiflagellates. And that was quite surprising. That came about from Nicole King's 2008 when the lab sequenced another quantiflagellate, Monoceca brevicalis. It's not clear, at this point, I don't know whether or not we can reconstruct exactly the events at which they happened, but I think what's interesting from my perspective is what could they have potentially been doing in a unicellular context. So we can kind of make inferences there about how they might have been co-opted. And so, for example, in the eLife paper that we published last year, we find that perhaps interactions with the environment, and more specifically interactions with bacteria, might have really driven some of those the evolution of those pathways.
That was actually going to be my next question, which is which of the molecules or particles which are now being used for this process were co-opted, and which had to evolve separately to make it happen better. But what were they actually doing upstream of being used for these things? Do you see any of the processes or proteins having double roles, which indicates that they may have been stolen from another process to enable cells to stick together like this?
Well, I think certainly one thing that's a problem is that there's such a large gap in the fossil record that we can make inferences, and certainly by studying the biology of quantiflagellates, that give us a pretty good handle of the realm of possibilities that these proteins might have been originally used for. So, for example, things like the proteins involved in our innate immune system may certainly have been co-opted from interactions with bacteria on a normal basis. And so that's an instance where some proteins now in the immune system might be used to both sense pathogen as well as to sense things like commensal bacteria and be able to gauge an appropriate response.
Thank you, Rosie. That's Rosie Alegado. Let's go back to viruses again, because joining us from Beijing, Wenhui Li, via a slightly difficult set of telecommunications hurdles, but is coming to us via a phone and then the computer. Hello and welcome to you. Tell us a bit about you. I'm not hearing anything. I don't know if anyone else is.
Hello, everyone.
Hi.
Yeah.
Hello. Good to have you with us.
I'm Freddie. My lab
Tell us a bit about you and your work.
Yes. My lab is working on viral infections of hepatitis B and D viruses. They are really fascinating viruses. We use virus biology and chemistry approaches to understanding these viruses. We have made some research progresses after we published our paper in eLife. And so we of the receptor studies of actually
the piece of work that you published there's a huge delay, so I'll ask this question and then hopefully you'll get it in a second. The piece of research that you published was groundbreaking because you managed to find the receptor, the way in for hepatitis B getting into liver cells, which people had tried and failed to do for many, many years. First of all, what is that receptor? And then tell us how you did it.
Oh, yes. Yes, the receptor call is sodium taurocholate co-transporting polypeptide. Yes, it was indeed very difficult to identify the receptor. Actually, we identified it by taking a new affinity-based purification approach. That is, we employed a unique zero-distance photo cross-linker and tandem affinity purification strategies in combination of high-end mass spectrometry analysis with the aid of a high-quality transcriptome database primer peer hypothesis developed in the lab. So it's a biochemistry approach, and we feel we are lucky we choose to peer at our animal for identify the receptor. And we succeed.
So just to summarize, because having read the paper, I was very impressed indeed. I mean, it's an incredible piece of work, because what you effectively did was to build artificial proteins which had this UV sensitive cross-linking ability, and then to test relentlessly all these different preparations against virus antigen to see which proteins appeared to be stuck to it, and then you had to work out which proteins they were in relation to the cell, and then which gene encoded them, and then work out what the protein was ultimately, and you came up with this entry receptor. How has that moved the field forward now? Now that we know how the virus is getting in, how has that changed things?
Oh yeah. Yeah, so with the discovery of the receptor, studies of HPV basic biology and antiviral therapies now can move faster, much faster than ever before. Actually, I'm very glad that our work has been well received by our colleagues in the field. The pharmaceutical companies also began to use the receptor expression cells to do drug screenings and development studies. I think it will be great that our research work may eventually help to change some people's lives in the future. So we also have seen some important new findings, some by ourselves and some made by our colleagues around the world.
The sodium taurocolate co-transporting polypeptide, which is this entry receptor, that actually had been blocked by other drugs in the past, hadn't it? So in what way can you now use the knowledge of this as the entry receptor for Hep B to find a drug that's cleaner?
That's a good question. Yeah, so basically our cells complement with the receptor will provide a platform to discover drugs, not only against the receptor itself, but also to other targets that is possible.
Yes, because one problem is that these drugs don't just block hepatitis B, they're going to block other fundamental processes that the liver cells and perhaps other cells need in order to remain viable or for a person to be healthy. So is there any way that you can discreetly block the virus entry without completely screwing up the cells?
Yes, we are working on this. Actually, we identified some fragment, some site on the receptor which can differentiate the viral entry versus the bite salt transporting. So we think that part could be a good target for blocking viral entry. So you're right, and actually the virus and the bite salt transporting can interfere each other for using the receptor. Some other colleagues in the field have actually very recently further found that the infection of HPV may interfere with bile salt cycling and the liver metabolism in general. So our receptor may be directly involved in the pathogenesis of a viral infection. So we are excited about these findings because they may help to our understanding of the pathogenesis of hepatitis B.
Is it not possible to deluge someone's bloodstream with some kind of decoy protein which looks a bit like or is a form of the sodium taurocolate co-transporting polypeptides, so that it basically soaks up all the virus and it therefore can't go into the liver cells?
It's difficult to use fragments from the receptor. We tried, but we have not succeeded. But the other way is to use the antibodies to just attack, to neutralize the virus directly. Our colleagues here in NIPS have found such antibodies. So I think that's a good way to block the viral infection.
Thank you very much, Wenhui Li. And as I say, a very impressive paper, because how long did that take you to do? Was it 5-6 years of relentless purifying those proteins that you cross-linked? It must have been a few years at least, wasn't it?
Yeah, we actually spent several years. Before we succeed, we tried other approaches, and we built up our toolbox during these past years, and that helps us a lot.
Indeed, but congratulations to you and the other two researchers we've already spoken with, Rosie and Jessie. Let's go back to Jessie, because we've been talking about a virus, we might as well carry on talking about another one. So let's return to your field of flu. What do you think the big priorities to try and solve in your area are now?
Well, I mean, that's a tough question. I mean, I think that from a medical standpoint, obviously, the goal that we have for all viruses is that we could somehow make an extremely effective vaccine to completely control or eradicate the virus, like was done for smallpox. I think, at least for flu, that's an extremely challenging goal, because the virus is found not only in humans, but it's also found in a very large number of other hosts. So it's hard to imagine eradicating the virus from all of them, and the virus is very adept at moving from one host to another. So, to really prevent it from being in humans, you would probably have to eradicate it from. But there's certainly some very exciting work going on that could hopefully make influenza vaccines better and broader. So I think that's one area of a lot of interest. Another area of interest, just from an evolutionary angle, is really, I believe, using sort of high throughput techniques to better understand how viruses evolve. So this is less of an applied question, but I think it has a lot of importance for our understanding of these basic phenomenon. And what's really become possible is, it used to be that you sort of, if you wanted to study a viral mutant, you made that mutant, you studied it. You made another mutant, you studied it. And what's really become possible now is you can make large numbers of mutants at once, and you can use high throughput techniques like next generation sequencing to really characterize all of the mutants that are there and really get a sense of what the sort of local evolutionary neighborhood around a particular sequence or strain looks like. So in my mind, one of the more exciting areas from sort of a basic evolutionary standpoint related to influenza is sort of how can we use these techniques to better understand how the virus evolves, and how can we use these techniques to see what's actually going on during real viral infections. So we know that when an individual is infected with viruses, there are actually lots and lots of different viral variants there, and until recently we couldn't really look at that very well. We could just kind of look at the consensus, or kind of the average virus, and it's not always clear that that average virus recapitulates all of the properties of what's actually there. So in my mind, a lot of the most exciting areas from sort of understanding the evolution of flu is really using these new techniques to just generate a lot more information, which we then have to figure out how to analyze and relate to the questions of interest.
Because I was going to then follow up by asking you, and you sort of hinted at this, to what extent are the relationships between one mutation begetting another that you're seeing dependent on the cell type or the animal type that the virus is replicating in? So if I put the virus in a different context, or if I put the mutation in a different context, it's not in flu anymore, it's in a different, say, virus, or maybe even in a bacterium or a eukaryotic cell, do those same relationships that you're seeing occur, or are they only true in very specific circumstances?
This is a huge question, and this is sort of, really the motivation for example, our study in eLife is really to sort of address this question, because what we always like to do in science as much as possible is generalize. We always like to believe that we're studying one thing and it's going to tell us something which will not only answer the very specific thing that we're studying, but will also have some generality beyond, as you said, the specific viral strain that we used and the specific cell line that we used and what buffer we were using and all sorts of things like that. So we'd like to really be able to generalize these things from an evolutionary standpoint. What that means is you'd like to be able to do an experiment on one viral strain and learn something maybe very specific about that. But usually, really, the reason you're doing that is you'd like to think that there's something you can get out and you can generalize. So, so I guess you asked a couple of questions there. So first of all, one of the things we would just focus on is to what degree when we study mutations in one particular strain of influenza, to what degree are those mutations going to have the same effects in different strains of influenza? And that's really what we've been directly studying. And we think that we've identified certainly several mechanisms by which a mutation can have different effects in different strains, but we think there's also a lot of these mechanisms are fairly conserved. So for example, for influenza, we found that among the types of mutations that actually fix during natural evolution, the primary ways that those mutations seem to interact is that some of them would make this protein more stable and some of them would make this protein less stable, and sort of depending on the balance of these things, the protein would either fold properly or it wouldn't. I believe that that's probably going to be a fairly general feature of the evolution of a lot of proteins. Again, you can never really say that without doing additional experimentation, but I think a lot of that really comes down to the fundamentals of what most genes do, which is encode proteins, and then what most proteins do, which is to fold to some sort of structure. And I think that's what sort of a lot of the evolutionary selection is actually for. Obviously, there are other things, more subtle things layered on top of that. But yeah, I mean, so I believe that we can probably extract some generalities from studying one particular viral strain or one particular viral protein and extrapolate those to other situations, but it's certainly not going to be perfect. And related to that, I would say that one of the things that we really hope to do is sort of develop better models for how we think the evolution should go, not to be predictive, but to allow us to sort of tell when we're wrong. So in evolutionary analyses, usually the most interesting thing we do when we look at gene sequences is find something that sort of looks unusual. So someone, for example, if you look at a gene and you can see that there's been a lot of recent evolution, that sort of looks unusual because most genes change slowly, and in those situations, a lot of times there's something interesting going on there that you follow up. And I think as we develop more and more mechanistic and detailed pictures of how we expect evolution to proceed, we'll get better at figuring out when there are things happening that we don't understand, and a lot of times those are the most interesting things to follow up on.
But there must be situations where if a protein gets bent in one direction and it then develops a compensatory bend in the other direction to straighten things out, there must be lots of different ways that you could arrive again at the same sort of straight protein with kinks in different compensatory places. Rosie?
Oh.
I thought you were going to say something. Sorry,
no.
Go back. Sorry, Jesse, go ahead.
What I was just going to say is, I mean, that can happen, but a lot of work going back decades now is one of the things that's been observed in the evolution of protein coding genes is that protein structures are very highly conserved relative to the genes that encode them. So, if you have two genes that have only 50% sequence identity, they may or may not be doing exactly the same function. There may be a lot of differences between them, but they're probably folding to very similar protein structures. So I wouldn't say that sort of the structure bending in different ways is really the right way to think about it. All these structures do change a little bit. They're fairly conserved over evolution. But certainly there are a lot of mutations that can make a protein more stable, and there are a lot of mutations that can make a protein less stable. So there's a huge amount of potential for interactions among these mutations. And that's, I think that's definitely true, and that's why it's hard to ever be predictive. It's not, when you go back to the very early study of sort of genes and diseases and mutations, Linus Pauling characterized sickle cell anemia as this disease where you have this one specific mutation, and this one specific mutation causes this very specific phenotype, which is sickling of the red blood cell. And probably most mutations aren't that simple. Probably most mutations have a lot of context dependence. They'll have an effect that will depend on other things that happen. There's a lot of complexity there. So I think dealing with that is one of the really big challenges of biology.
Rosie, do you see any of the same sorts of trends emerging in the relationships you're trying to study and the conversations that bugs have with your cells?
Well, I would say we're really at the beginning of kind of exploring what that might be. So, for example, one of the things that I'm really interested in my lab is to understand what the molecule that I identified that interacts with the choanoflagellate, whether or not there's like a broader biological context for that. And I think that's a little bit similar to what Jesse was saying. Is we all want to know what the broader significance is. And so, for example, the molecule that I identified, which is a bacterial sulfonolipid, which structurally looks very similar to sphingolipids, which are secondary messengers. One of the questions I have is to see whether or not this might be just the first identified molecule in a class of bacterial molecules that might be signaling to animal cells or other cells in general, and that you just identified perhaps the founding member of this class of molecules. So, for example, the bacteria that makes this molecule is a member of the Bacteroidetes phylum, and those bacteria are dominant members. They predominate in the animal gut, and so one hypothesis that we're currently trying to explore in my lab is to see whether or not other members of this group produce molecules, similar classes of molecules, and whether or not those molecules are also bioactive. And so I guess in a similar vein, that might be our approach to kind of getting at that general question.
Because we're suddenly beginning to wake up to the fact that these sorts of conversations that go on between microbes and host cells are terrifically important. Our entire immune system fabric is structured according to probably very early interactions we have with our microbiome. How healthy we are comes down to the spectrum of bugs that live in our guts and on our skin. And so really there must be some insights coming from what you study which informs those bigger picture type conversations that go on at the molecular level.
I think one of the biggest insights to me personally was the idea that you had these very early interactions going on, and first of all, they are rampant now in all existing animals, but just the idea that these kinds of conversations and these long-standing relationships persisted all throughout our evolutionary history, and perhaps might even have been responsible for selecting for multicellularity or providing that advantage is very provoking, and indeed really brings home to me the centrality of microorganisms to coevolutionary processes. And I think one thing that has also really struck me is the idea, and that's why I say my work is motivated by the idea that everything, pretty much all life, has evolved in a sea of microorganisms. Is that when we think about evolutionary developmental biology, a lot of times we're thinking about the organism, but in fact there's a rich milieu of chemicals from the external environment that have influenced and impinged upon their biology. So I think that that's something that is becoming more apparent.
What about the conversations that go on not between microbes and their hosts so much as between microbes?
Yeah, certainly I think that is a very, very rich area of study as well. And so, for example, the molecule that I study, the sulfonolipid, it's very hydrophobic. And so a second question that my lab is really interested in is how can a really hydrophobic molecule, which presumably when it's released into this aqueous solution, how can it get to something like a choanoflagellate? And my hypothesis was that it perhaps was being packaged in outer membrane vesicles, and that outer membrane vesicles, which are produced constitutively from the outer membranes of gram-negative bacteria, might be a delivery method for this molecule as well as more generally for other hydrophobic molecules. And outer membrane vesicles are really, really interesting because they can not only serve as little packets that traffic back and forth DNA, proteins, toxins, but they're the kind of molecules that you don't need high specificity between cell types. And so these outer membrane vesicles potentially could be a way that you can have signaling not only between intra-species conversations and interspecies of conversations between different groups of bacteria, but perhaps even inter-kingdom type of conversations, because they're just kind of like these little lipid packets that are sent out there into the environment. And so I think that potentially the work that we started in eLife might lay some foundation for us to begin to look at different kinds of conversations between bacteria and different classes of molecules, specifically lipids, which has been so hard to kind of approach previously.
Indeed, there was a paper which was published in eLife just in the last couple of months, looking at the interactions between a Vibrio species standardized laboratory model now, isn't it, of how these particular Vibrios can induce the squid to allow the bacterium to colonize specific pockets that then ultimately lead to the production of light on the surface of the organism. And we're now hearing that perhaps even LPS, the thing which we all dread as a cause of septic shock and is linked to septicemic disease, that this is actually a signal. So under certain circumstances it's pathogenic, and under other circumstances it's part of the conversation. So it goes back to what you're saying about co-opting other molecules to do a range of different jobs.
That's right, and I think that I really like that you brought up the Vibrio model, because for a long time there was really a dearth in model systems where we could really look at the evolution of host microbe interactions, and certainly the Vibrio model has led the way in terms of reshaping our understanding of how bacterial molecules that we may have originally thought of as really only being involved in immune type processes really have much more basic roles. And so I think that's also one of the strengths of the quantiflagellate system, is that we can really approach that from a very basic point of view. And actually, my background is in bacterial pathogenesis, and the motivation for me kind of getting into quantiflagellate microbial ecology and joining Nicole's lab was to get into that even more basic role, is to kind of, pathogenesis wasn't it and all of the story, and to kind of really look and peel back those layers and see how evolutionarily how these conversations between bacteria and their hosts might have evolved, and microorganisms even broader.
Well said. Let's talk hepatitis B again for a second with Wenhui Li. We've heard from everyone else how their bug that they're interested in talks to its host and talks to its neighbours. Why do you think hepatitis B latched onto in the first place using sodium taracolate co-transporting polypeptides as its entry receptor? Why was that a good choice from an evolutionary point of view for hepatitis B, do you think? I'm hoping the message got relayed okay.
Can you just repeat?
Have we lost him?
Can you repeat the question for him, Chris?
Yes. Sure. Why do we think that hepatitis B used or evolved to use the sodium taracolate co-transporting polypeptide part? Did it evolve to do that?
Wow, it's a very difficult question to answer. We don't know now. We don't know the answer now, and but the NTCP, the receptor, is kind of abundant on the side, on the blood side of the hepatocyte. So it's convenient for the virus to use this receptor. And I think it's just from evolution selection. So, as you know, that in biology, nothing makes sense but in the light of the evolution force. I think it's the HBV and HDV happen to select NTCP as is a receptor. But there are some underlying mechanisms we are trying to understand, but at this moment, we don't know exactly why.
One of the interesting thing about hepatitis B is its ability to persist for extended periods in its host. To what extent the use of this particular entry receptor, where it goes and how long it stays in those cells?
That's a very interesting question. We think that the entry must be linked to the establishment of a chronic infection. But at this moment, we don't know exactly the mechanism. I think there must be some link. But now we don't know, to be frank, and we are actually working on this.
Oh, good. Well, if it culminates in another paper in eLife, perhaps you can pop me into the acknowledgments. Thank you very much. Thank you to everybody, actually. We're almost out of time. So thank you to Jessie Bloom, also to Rosie Alegado. Rosie, you moved to Hawaii. You said that you thought the weather would be better over there. Is it?
Absolutely. It's great.
Have you been surfing?
You in Berkeley wasn't that bad to begin with.
Berkeley was not that bad, but I have to say, I mean, I'm originally from Hawaii, so Hawaii is much better.
Okay, well, when are you going to invite us over?
You can come over anytime. That would be awesome. All of you guys.
Thank you very much. There's an invite, and everyone who's the audience to this hangout, of course, is invited over to Rosie's place. Thank you very much, Rosie.
No.
Right, well, we are out of time. So we have to say thank you very much to the three of you. You did a brilliant job, sort of technology, to sometimes make it work, and you did brilliantly, so thank you very much. And of course, thank you to eLife for coming up with the idea of doing this. I think this is going to be the first of a number of these things. Now, I've got to make an announcement to say that eLife is ready to announce the next round in eLife sponsored presentations, and here are the people who are going to be participating. Curtis Huttenhauer is from Harvard School of Public Health, and the paper that Curtis is going to discuss is expansion of intestinal Prevotella copri correlating with enhanced susceptibility to arthritis. In other words, the link between carriage of a certain microorganism and your arthritis risk. That was a really striking finding that got quite a bit of attention. Also, Roberto Keller is from the Gulbenkian in Portugal, and he and a colleague made a dramatic finding that they could find out, if you look at the structure of the thorax in an ant, you can find out what sort of ant species it is just by looking at how the neck has evolved of this ant, whether or not it's got big muscles that it can then eat away and turn into energy to feed its colony if it's a queen, or go out and go and get food if it can't do that. So he'll talk about that. Also, Stephen Baker is a Wellcome Trust Major Overseas Programme Fellow, and they have made a very, very surprising finding, actually looking at fluoroquinolone-resistant Salmonella typhus. Normally, when bugs evolve to become resistant to antibiotics, there's some kind of fitness cost. They become less reproductively fit. He told me that when his PhD student first presented him with the data that the bugs that they were making resistant to these organisms were fitter than their parents, he said, we just didn't believe it, and we did it 10 times. So a very interesting story emerging there. Also, Rajat Rohatgi, who's from Stanford, is going to talk about structure and function of the smoothened extracellular domain in vertebrate hedgehog signaling. Rajat's work has involved investigating the role of oxysterols in the signaling pathways of embryo. We'll also hear from Matthias Malaguti, who's at the University of Edinburgh, and works on bone morphogenetic protein signaling, which can suppress the differentiation of pluripotent cells through E-cadherins. So that's all coming up. I should also point out, I've been asked to say, the page, I'll find the right line. An announcement is going to be made tomorrow. Look at the eLife site for details. Congratulations to these authors. So look at the eLife site for a big announcement tomorrow. But thank you everybody for taking part. It's been great fun, and I've learned a huge amount. Goodbye.
Bye.
Bye.